And welcome to this session on flash mechanism and with a physics angle. It's my privilege today to introduce our distinguished speaker, Professor Schuemann from MGH in Boston, who will give this keynote lecture on modeling the mechanism of flash effect.
Right, thank you very much for the introduction, and thank you very much for inviting me to give this presentation about the physics mechanism of flash. I did feel a little bit like having to talk about giving you a short story about why the Earth is flat, so I'm going to try to do that in the next couple of minutes. First of all, I have no significant conflicts of interest except for some grant funding. I'd like to start by thanking a lot of people, especially the FRPT team, to make this conference so successful in such a short time in virtual, from going to in-person and virtual. It was impressive. The MGH Flash team, where a large portion of that is depicted on the right, that makes me be able to do all these fun experiments. Shannon Thompson and Stephen McMahon for sharing some of their slides, of course, funding agencies for making all the work possible, and really everyone that's working on Flash, because this is a fascinating field and so multidisciplinary, it's just fun to work with. And we need to know a lot more. So the physics mechanism of Flash can be summarized quite quickly. I don't think there are any. So thank you very much for this attending this talk. And if you have questions, please post them. But of course, we can talk a little bit about the actual mechanisms of modeling the mechanisms of Flash. So we kind of changed the title a little bit from just the physics mechanisms to modeling mechanisms. And I'd like to apologize in the beginning that I will not include all models that are available or published. I'm trying to attempt here to outline a few different modeling approaches that are based, but I believe, in a physics kind of approach and or based on some mathematical modeling. Oops. So for me, we start typically with track structure Monte Carlo models, a very nice bottom-up approach where we really model the detailed interactions at the nanometer scale, then try to include more and more details, including the chemistry, some biological modeling. And if you ask a physicist, we typically, well, sometimes we say that we should really be able to describe the entire world just with physics equations. So that's an ideal case. Of course, there's a but the world is complex. And so what we typically do in track structure Monte Carlo approaches is we separate the time into certain sections. We say the physics time is typically where we have track structure that we can describe with track structure in the first femtoseconds. Then there's the physical-chemical stage where we have the creation of water, we have water radiolysis and creation of some reactive oxygen species. Then we have the chemistry, where these species actually can react with each other and migrate. And then we have biology taking over some time in the area somewhere between nanoseconds to microseconds or milliseconds, where biology then repair of the damage that was induced in the initial stages takes over and tries to, again, repair this damage, where repair processes and failures can come in, which of course can be controlled by or impacted by the microenvironment and results in some observable biological effect in the end. And so that's the damage repair side. And flash irradiations typically take place in an area either somewhere between a few microseconds maybe to some milliseconds. And conventional dose irradiations obviously are typically in the area of seconds to minutes. And so you can see that the area in between here is typically where we focus on. That's why we focus on mostly chemical and biology modeling or understanding the mechanism in this time frame. And the track structure Monte Carlo codes can be nicely depicted by just looking at a single track. So you can see here is a DNA plasmid and you can see the track coming in here with the proton coming in with secondary electrons, and then you can see the creation of radiolysis species. And then once chemistry starts, you can migrate these reactive species around, and this is pure water. And you can see that we can have effects quite a distance away from the initial track. You can also see that the track here is in the picosecond range, whereas then the chemistry starts in a nanosecond, and typically by this time, all the species would have been scavenged already in a cell environment. And so once you have this done, once you have the chemical species process, you can go into biological modeling. Here's a part of a model that is developed by the Manchester group that has now been included in Topaz and Bio, that has a very nice way of modeling all the different repair processes by recruiting different repair proteins and having probabilities of different processes that happen and result in either repair or misrepair. There are, of course, a few caveats of track structure models. It can describe a lot of things, but it is quite good to understand the limits to it. And one of the limits is just that we don't really know our physics cross-sections, already the physics cross-sections. We don't know, even for water, at these low energies that typically are necessary, to a very accurate amount. So we have some 5 to maybe 10, 20% uncertainty there. We have chemistry that is mostly also only done for water. The cross-sections are also mostly just for water. We're working on, or we're already including more and more scavenging and microenvironment factors, especially for the chemistry. So this is good. But of course, at some point, the track structure codes just stop in what they can really model. And then we start to have all these intercell interactions and other biological effects, maybe long distance effects that are currently not at all considered in these models typically. So there's a large uncertainty if you want to really get to absolute predictions of effect sizes, but these models are typically very good at modeling relative effects. So let's look a little bit more at this. So there's the physics is basically, we have the physics part is very short. So in 1 picosecond, you would traverse many cells with a single track. So the track cores are typically in the order of a microsecond. And if you look at a track, a single proton track here, you would have a few hundred, probably, proton tracks per nucleus, per gray, that traverse your nucleus. So you have a couple of these tracks in a maybe 10 nanometer, micrometer nucleus roughly, and you can then see how likely it is to overlap these. Then if you go to higher and higher LET, you can see you will reduce these longer ranging electrons and get to more of a track core, but the track core will be getting bigger. So these effects kind of come into play if you want to look at physics. But so if you want to really look at physics interactions, you would really need a single shot delivering all the protons at a single time because of the short time scales that we're looking at. So this could be potentially laser accelerated or kinetron particles that you can deliver. But most flash irradiations really are at the millisecond time scale. And so for physics interactions, that really separates them far in time, and so even geometrically there would be no overlap. But we can include the chemistry and calculate the geometrical overlap for this. The group of McMahon, Stephen and Shannon did this interesting study where they looked at the development of the chemical species and then estimated roughly 80% area where 80% of the chemical reactions or species are created. And then you can look at this and define the radius of this track core, depending on how long you let the chemical species migrate. Of course, the longer time you give them, the further they migrate. And then also depending on the particle track. So in the LET, which you have increasing radius obviously with the time and decreasing with LET. And then you can do a simple geometrical analysis of how likely is it that you get an overlap in a cell nucleus. And that's exactly what they did with a simulation of single pulse, assuming single pulse laser irradiation, so very short time frame. And you can see here, if you go down to a microsecond, you can get actually quite significant overlap. But in a cell environment, we are typically more in the nanosecond range, so maybe 1 to 10 nanosecond, and by that time, all the species would probably be scavenged. And so we are really in this area down here, where the difference between them is not very big at all, and you have a maybe 5–10% probability of having some overlap of tracks. This is, of course, for 2 gray. If you increase those, this probability greatly increases. So at 10 gray, depending on your LET, you are somewhere between maybe less than 5 to 50% that you have a chance of overlap within this chemistry core, and then increasing from there on. But within biological timeframes, these intertrack events are unlikely to play really a significant role in clinical outcome, especially if you consider that this is simulations for a single pulse irradiation, and most FLASH simulations actually take some time. So Jose Ramos did a very interesting study where he actually used the track structure code to simulate intertrack particle interactions, and looking at different beam pulse deliveries of somewhere between 1 nanosecond to 10 microsecond. These are actually randomly distributed in time, but it looks a little skewed due to the logarithmic timescale on the bottom. And you can see that there is an effect on the different on the number of species that are created, but mostly in when they are created. If you look at past 10 microsecond, you basically have more or less the same curve, and this is for 1 MeV protons, 100 MeV protons, if I remember right. And you can see there's some effect on the G value of different species. This is simulations including scavenging, so this is a very nice study, and you can see, but the overall, the effect on G values, I believe this one is also for OH radicals. You can see the scale of the effect is somewhere in this shaded area, where this shaded area really is the definition of assuming all tracks are being delivered instantaneously and all tracks are delivered independently, so at a long time scale. And so you should be somewhere within there, depending on your actual delivery. So there's a little bit of a potential for inducing some flash effect or some differences due to the different pulse structures. Boscolo et al. Also did a very interesting study using the TRACS chem code. It's basically very similar. They looked at the water radiolysis and followed the tracks over time, and the G value estimation after. So they one of the caveats is, again, that is a water simulation, but it's consistent with the measurements of oxygen depletion in water. And they very interestingly looked at the effect on survival in compared to experimental data. And you can see here is the gray shaded areas, actually the difference between their predicted flash and conventional dose rate irradiations. And so this difference cannot explain the difference between the observed, the red line and the blue flash effect that is seen in the sparing. So they conclude that there may be a role for oxygen depletion in flash, but it's probably not the most significant effect to induce flash sparing. Hongyi Zhu did a very interesting macroscopic study on oxygen depletion, where she looked at data from Vopel et al. That had actually here aggregate measured the oxygen tension in different patients for normal breast and breast cancer, and here for 2 specific breast cancer patients. And then if you assume some parameters for your oxygen depletion, you can get a depletion of oxygen. And she did an actually very nice model where the depletion and the deoxygenation, or the oxygen depletion and the damage induction, actually go hand in hand. And so you can see over time, you then get a shift in the probability of your cells to survive, with obviously the more hypoxic cells surviving longer. or having a more significant, remaining more in your cell sample. So, however, if you look at this from look at the different oxygen distributions, you will see that for most cases, you will actually get a much more significant effect for the breast cancer patient than you would get for the normal tissue. Mostly because for most breast cancer patients, they have an average much lower oxygen tension in the tumor than they have in the normal breast. And you can see here for these triangles, they are 10 gray. So at 10 gray, you have much higher flash RBE, as she calls it, than you have for normal tissue. Of course, you can argue this, and you can see that you need to go to a little bit higher doses to really induce some kind of a flash sparing effect, because you need to have significant shifting of this oxygen concentration. But you can argue, well, maybe it's not that the average concentration matters, but that the tissue that really determines the control of tumor is the already hypoxic tissue. So in that case, obviously the shift in oxygen concentration for the tumor may not matter at all, and you still may get a sparing effect from the flash. So it's a very interesting study to look at different parameters. She also had a study where she looked at the NASIC code, looking at track structure again to calculate the radiologic oxygen depletion, but also adding a mathematical model for looking at the oxygen tension and recovery from nearby vessels. And so you can see, depending on where you are and how close the vessels are, you have different recovery times, and you combine that to then get these very interesting patterns where you have potentially your initial oxygen depletion and then the recovery. And if you have a single pulse, you get a much more pronounced peak. But interestingly, if you have this pulsed radiation structure here with certain recovery parameters, you potentially might get a longer depletion of oxygen in your tissue. However, in either case, the oxygen depletion is not incredibly significant. You can see here you have sometimes up to maybe, well, in single pulses, some significant oxygen depletion, but you're not going down to really hypoxic areas. So you still need her conclusions, you still need low LET and low oxygen concentration as initial condition to have a sizable effect. And similarly, the group from Manchester, from Rothwell et al., looked at a really interesting study looking at the correlation of 8 different parameters. So look at the correlation space here and looked at how the different parameters impact each other and what parameters combinations are necessary to likely have a flash effect. And their conclusions are you probably need somewhere in the order of tens of grays per second dose rate. You have sufficiently high dose and, again, slight hypoxic tissue or low oxygen concentration in your initial tissue. And you can really move these around and see how all these parameters play together. So it's very interesting. But it's they show that how the sparing is potentially dependent on multiple parameters here and how they interact with each other. So I talked a lot about oxygen depletion so far. The main reason for this is oxygen depletion is actually something that feels inherently like it makes sense. It's easy to understand. It's a mechanism that we know how to model. And so this is why a lot of studies actually look at oxygen depletion as a mechanism of flash. Of course, there's also some questions about this. First of all, it's hard to measure exact oxygen concentrations in tissue at microscopic scales. And the question then is, what scale is really, really necessary? But also, oxygen depletion is supported by some data, but other data really suggest that oxygen depletion may only play a minor role in the flash sparing effect. So while modelers really like oxygen depletion, the verdict is still out if this even is a mechanism that contributes to flash or is dominant in the flash sparing effect. There are also a couple of unknown factors. we don't really know, as I said, the oxygen levels in vivo, especially across tissues at subtypical voxel level resolution. Also, the question is, what scale is really important? Is it the tissue scale, the cell scale, or do we need to go down to the subcellular compartments? And what are the targets that we really are looking at? Is it just DNA damage that we impact by having the oxygen enhancement ratio, or is it other things like mitochondria or even the cell membrane that have been thought to play a role in flash tissue sparing? So it's important to think about where do we measure and what do we really measure when we measure things. So if you measure a typical PET oxygen distribution, you're averaging over quite a large area there. And so we need to understand what is our resolution and are these measurements really what is relevant to our biology? Because if you look at a cell, for example, Schwartz et al. Was having an interesting discussion at the Radiation Research Society meeting that just a few months ago, where the oxygen concentration actually also varies largely across a cell. And so if you say that your oxygen concentration at the nucleus is what really matters, or at the mitochondria, then these may be two completely different things. And if you really want to resolve these things, you may need very high resolution in both scale and in time to really understand what the different mechanisms are. And
3 minutes to
modeling is key. And so there is, of course, other models like not oxygen depletion, but oxygen related, that have been discussed in large parts. So different mechanisms here that can be also modeled with Monte Carlo. But since they go more and more into the chemistry that has already been discussed in large parts yesterday, and I will not go into the detail, I do like to point out another modeling approach that has nothing to do with oxygen or chemistry. It's the lymphocyte sparing, where we're looking at circulating blood. And so this is a kind of Markov chain model, a very simple one for a mouse. In a human, we have a slightly more complex model, but it basically is all different organs of a mouse, and the heart and the blood typically flows into the heart and then into different organs. And if you assume that for a flash irradiation, you have only the entire dose received by the blood that's actually in the target volume, whereas for conventional dose rate, your blood keeps flowing through, you can have actually significant portions of the blood irradiated. And if you see here with 2 gray, you can actually deplete a lot of the lymphocytes. And so you can model this. And if you use a 2 gray threshold, this would give you potentially a factor 3 for a 16 gray delivery, factor 3 sparing of the lymphocytes in the circulating blood. And so if the circulating lymphocytes in the blood are actually important for the flash effect, then you might see, this might explain that. There are similar models by Jin et al. Again, basically having the blood flow through the heart and either irradiated or non-irradiated volume. The major problem with these models is that so far they are not supported by any experimental data. Experimental data doesn't seem to show that circulating lymphocytes are what causes the flash effect. Now, just modeling the flash effect is something exciting, I think. The question is, what mechanisms are involved and what do we need to model? We probably need more than just physics. There's definitely an interplay between physics, chemistry, and biology, so it's really interdisciplinary. Modeling is important, I think, to really help us understand the underlying mechanisms. And if we really want to move this field forward into the clinic, I think it's very important to understand the mechanisms just so we can get the most out of our potential flash sparing effects. But it's important to think about what you are modeling. So in some scenarios, you may want to model more the cell or just pure water, but you have to be sure to model exactly what you're looking at. You also have different reactions that you want to look at. They define the time that you need to consider for your modeling. And then, of course, that impacts the time structure of your beam. And you may have to invoke a lot of different cell tissue or microenvironments, so different scales that may be important. Maybe it's a cell effect, maybe it's a tissue or other biological effect that may impact your flesh or is responsible for the flesh tissue sparing. But overall, it's a great opportunity, I think, for really a multidisciplinary research. And maybe with some modeling and experimental guidance, we can really find new mechanisms of radiation actions. So thank you very much for listening, and please do ask your questions.
Well, great. Thank you for this very interesting talk. I don't know if we have some question already, but at least we have a little bit of time for questions. I don't see it in the chat. Do you see it? Here we have. There is a question. I didn't understand why the overlap probability for laser plasma accelerated proton is increasing with longer scales.
Oh, that is.
It's counterintuitive.
Sorry, that's not the time scale of the actual laser delivery. It's the time scale that you model the chemistry. So if you allow your track core of chemistry to basically model it for longer, if your species have longer time to diffuse, then they can basically reach a larger space. So your track core gets bigger. Of course, in a cell environment, you will never really get to a microsecond for most reactive species. So typically, you're more in the nanosecond area.
Okay.
And yes, the density
another
is higher
question
than
here. Jan, thank you for this great talk. With TOPAZ NBO include the flash oxygen depletion simulation function. What do you think?
Yes, yes, it does include it now, but only in our not released version yet. We are working on that. That will probably come early next year. It's all working, but our chemistry experts are very good at wanting to make sure that things really work before they release it. So we're working on it to release it.
But Jan, I have a question. Let's imagine depletion, oxygen depletion is central in the flash effect. It probably would be more affecting the tumors and make them hypoxic and resistant. This is not what we see, in fact.
That is true. That is what the models show as well. But as I said, if you believe that, I mean, yes, in most standard simulations, you would say you have more hypoxic tumor, you get faster to a hypoxic region and therefore spare the tumor much more. But if you say your tumor control is actually controlled by the already hypoxic part of the tumor, then making that part more hypoxic doesn't really change anything. And so you don't really change the response of the tumor. So that may be what is important there.
Okay. If you would have the possibility to try to increase the flash effect, would you go to delivering more in nanoseconds or in microseconds, the beam? From your modeling, what do you think?
Well,
which direction?
Probably, it's a good question. Depends on the mechanism that you're actually looking at.
Yes.
I would say probably the faster, the better, just from what our experimental data gives us. The modeling part, you know, if you get too many tracks too close together, you can have a lot of interactions with the tracks that actually reduce the chemicals, the reactive species that are around. So, yeah, it depends a bit on the mechanism.
I think we have 1 minute left. Just short question, short answer, if we can. Is this about the lymphocytes? And I think there are two questions there. The lymphocyte cell death. What is the background for the difference between flash and conventional? And will it influence the immune response? That's a very exciting question.
Right. Well, that was the initial idea why we thought lymphocytes may be an interesting target to look at. And the idea really is that you have a much smaller amount of circulating lymphocytes that you irradiate with flash. All of the ones in the target field will receive the full dose, so those will be taken out or killed. But with conventional dose, right, you circulate blood through the target. For mice, for example, for 2-minute irradiation, you have all the blood going through the field multiple times. And so then you can potentially deplete all of it, circulating lymphocytes. The problem is that lymphocytes in the circulating blood are only like 5%, so you probably have a larger effect from other organs.
Okay, thank you very much again for this beautiful presentation. I think it's really time now to close the session. I would like to thank everyone for attending, and we go to the next session now. Thank you again.
Thank you.